This is a short lecture to show you the intricacies of DNA replication. So DNA is double-stranded, 5' to 3' on one strand, 3' to 5' on the other, held together by hydrogen bonds between the bases. When DNA is going to replicate, we have these places on DNA called origins of replication, or ORI, that open up. And there's going to be multiple origins of replications unless you are a prokaryotic cell. And so let's zoom into one of these origins of replication. So what's going to happen at each one of these ORIs, where it is single-stranded, you're going to get single-stranded binding proteins attached to the DNA. And then there is an enzyme called Helicase, which breaks the hydrogen bonds and unzips the DNA double-strandedness. So what's holding the bonds together are your hydrogen bonds, holds the nucleotides together, and we're going to remove them. Notice we have a replication fork, that's what this is called, on both sides of our original bubble. So the next step is, before we can even start replication, we need to add a few nucleotides because DNA polymerase needs a 3' hydroxyl to work off. So there is an enzyme called primase, which adds in a few RNA primers, complementary pairing to the DNA. And so if this is our 5' to 3' strand, you can see that the primers are going to be added in this direction, and I have my 3' hydroxyl group. Same thing happening on the bottom of the strand, except now the primers are added and they're going to be in the reverse direction. So here you can see my primers are added on, and now I'm going to be able to bring in my enzyme DNA polymerase. DNA polymerase adds one nucleotide at a time using complementary base pairing to the parental strand. So on this top strand in this direction, the DNA polymerase is going to bind over here and it's going to go in this direction. One nucleotide complementary pairing, and you keep making a bond between the hydroxyl end of on the 3' end of the sugar with the incoming 5' end of each new nucleotide. So here you can see my DNA polymerase on this strand is going towards the replication fork, and it's continuous. Once I've had that initial jump on, everything's going to just keep going. And same thing on the bottom strand. Here you can see I'm making my complementary DNA. So I've color-coded it that the new strand you can see matches the old strand, which would have been over here, complementary base pairing, and similarly on this side over here. Helicase is going to keep unzipping the replication fork, so that bubble is going to get bigger and bigger and more elongated. So as the helicase opens it up, this DNA polymerase is going to keep going continuously in this direction, and the same thing is going to be happening on the bottom strand in the other direction. We call this the leading strand, the new strand that is synthesized continuously always towards the replication fork. So you can see on this top strand, the leading strand started in this direction, and on the bottom strand, the leading strand is going in this direction. And you can see it just keeps filling it in. However, when we look at this, we've got a problem because we've got a gap forming on this strand and we've got a gap forming on this strand. So what has to happen here is we have to go back and fill in those gaps. So again, remember, DNA polymerase can only go in the 5' to 3' direction, which is the direction on the top strand, and on the bottom strand is going in this direction. So if I want to fill in this gap, I'm going to need to add some new nucleotides over here and then go in this direction, and you can see on the bottom strand it's going to have to come in this direction. So that's exactly what happens. More primers are added, and then DNA polymerase gets on and basically synthesizes and fills in that gap. But now we can see we still have a little bit of a problem because we have some RNA nucleotides, and we've got new DNA, a little piece of RNA, and then this leading strand that is continuous going in that direction. So what we have is we have a different DNA polymerase, but you don't have to worry about the details for that, which actually removes those RNA primers and replaces them with DNA nucleotides. So the RNA primers have been removed, and replaced with DNA, and these shorter fragments that we keep making are called Okazaki fragments. So we are going to have Okazaki fragments on the top strand going in this direction, and we're going to have an Okazaki fragment on the bottom strand going in this direction. Once the DNA polymerase has filled in those gaps, we still have a problem because we have a gap in the sugar phosphate backbone. And this is where another enzyme comes in known as DNA ligase. And the DNA ligase rejoins that gap, remakes that phosphodiester bond, and completes the DNA molecule. So now you can see this piece here is continuous going in this direction, and the bottom strand is continuous going in the opposite direction. Now remember, helicases keep opening up more at the replication fork on both sides. So this top strand will keep going, but we're going to create the same scenario in the bottom strand that we're going to need to fill in the gap. And we're going to have the same scenario on this top strand. We would need to add more pieces in and fill the gap. So this is just an expanded view where you can see on this top strand, Okazaki fragment, primers, Okazaki fragment, primers, Okazaki fragment, primers. And now our DNA polymerase would have come in, removed that, joined them together. Come in, remove that, join them together with the ligase. DNA polymerase removes the primers, puts in more nucleotides, and join them. So this newly synthesized strand is going to be much slower than a leading strand which is continuous. So these are often called the lagging strand, composed of Okazaki fragments that have to be joined together. And what you can see is this is happening in multiple of our bubbles, and eventually this bottom strand here is going to catch up with the next bubble. This bottom strand here is going to catch up with the next bubble. And this then makes the DNA completely synthesized. So we always have leading and lagging strands forming on both sides of our original parental strand. When they meet up, they are joined by ligase until replication is complete. So when replication is complete, we have two new identical DNA double helices. Each one has an original parental and a new daughter. So you can see that top strand new daughter matches the original parental on the other strand, which also has a new daughter, and this new daughter matches the original parental on the other strand. This process is known as semi-conservative replication.